Hey everyone, welcome back to the channel. Today, we're venturing into the mind-bending frontier of biocomputing with the groundbreaking work of scientists growing mini-human brains, also known as organoids, which are three-dimensional clusters of living brain cells derived from stem cells to power computers in ways that could slash energy use and supercharge artificial intelligence. If you're a tech enthusiast, a neuroscience fan, or just captivated by how lab-grown brain tissue is blurring the lines between biology and silicon to create more efficient machines, you're in for a fascinating exploration. Because this isn't science fiction. It's a real leap where tiny, living neural networks are being trained to perform computations, potentially revolutionizing everything from data processing to drug discovery by mimicking the brain's low-power efficiency. Pioneered by teams like those at FinalSpark in Switzerland, these organoid-based systems have shown they can learn tasks like pattern recognition faster and with far less electricity than traditional chips, opening doors to sustainable computing that could tackle climate challenges or accelerate medical breakthroughs. We'll delve into their extraordinary capabilities in detail, break down the technical process behind growing and connecting these mini brains, discuss their potential to reshape technology and ethics, and wrap up with the latest developments as of October 4th, 2025, including fresh trials and ethical debates lighting up the field. So, settle in and let's connect the neurons on everything you need to know about mini-human brains powering computers. The capabilities of these mini-human brains in computing are nothing short of revolutionary, serving as bioprocessors that leverage the inherent efficiency of biological neural networks to handle complex tasks like image classification or speech recognition with dramatically lower energy demands than silicon-based systems, potentially consuming thousands of times less power while achieving comparable or superior performance in adaptive learning scenarios. At their core, these organoids excel in neuromorphic computing, mimicking the brain's parallel processing, where billions of interconnected neurons fire in response to stimuli, allowing them to learn through electrical stimulation and chemical signals, much like a developing human brain, enabling applications from optimizing AI models to simulating neural diseases for faster drug testing. For instance, a network of 16 organoids can process visual data or solve puzzles by strengthening connections between cells, adapting in real time without the massive datasets required for machine learning, and their biological plasticity means they self-organize to handle errors or damage far better than rigid chips, making them ideal for resilient systems in remote or power-scarce environments like space exploration or disaster zones. In practical terms, imagine a cluster of these mini brains analyzing satellite imagery for climate patterns or predicting protein folds for new medicines, all while sipping just milliwatts of electricity compared to the kilowatts guzzled by supercomputers, turning what was once a lab curiosity into a sustainable powerhouse for the AI era. To grasp how this works, let's break down the technical process and mechanics. Because the science behind growing and interfacing these organoids is a masterful blend of stem cell biology and electrophysiology, creating living circuits that bridge the gap between wetware and hardware with precision that's equal parts art and engineering. The journey starts with pluripotent stem cells, which are versatile cells capable of differentiating into any body type, sourced from adult skin or blood, and reprogrammed in a lab to form neural progenitors that self-assemble into three-dimensional organoids about 4 millimeters across, containing up to 100,000 interconnected neurons after weeks of culture in nutrient-rich bioreactors, mimicking the brain's chemical environment. These mini-brains are then interfaced with multi-electrode arrays, thin chips with hundreds of tiny platinum electrodes that deliver electrical pulses to stimulate activity and read output signals, allowing researchers to train them like a neural network by rewarding correct responses with dopamine-like chemicals or electrical feedback, much like reinforcement learning in AI, but using biological synapses that strengthen through Hebbian plasticity. where cells that fire together wire together. A typical setup, like FinalSpark's neural platform, connects 16 organoids in a cloud-based array, where users send tasks via the internet. The organoids process inputs through their living circuits, outputting patterns of electrical spikes that algorithms decode into results, all powered by a low-voltage system drawing just microwatts per organoid, far less than a smartphone's processor. Operationally, the flow is elegant yet intricate. Stem cells are seeded into scaffolds, nurtured for 28 days, to form functional networks with spontaneous activity, wired to electrodes for input-output mapping, trained over hours with stimuli like light patterns for image tasks, and queried in real time for computations, with ethical safeguards like using non-viable cells to avoid sentience concerns. This makes organoid computing a hybrid marvel, where biology's efficiency meets silicon speed for breakthroughs in energy-scarce AI. The strategic implications of mini-human brains in computing are profound, positioning them as a bridge to sustainable, brain-like processors that could slash global data center energy use, currently rivaling aviation's carbon footprint, while accelerating fields like personalized medicine, where organoids simulate patient-specific neural responses to drugs, or climate modeling, where their parallel processing handles vast data sets without massive power draws. Ethically, they raise questions about consciousness in lab-grown tissue, prompting guidelines from bodies like the International Society for Stem Cell Research to limit viability. But their potential to democratize AI by reducing hardware costs could empower smaller nations or researchers in developing regions. In broader terms, this tech could spawn neuromorphic chips for edge computing in wearables or robots, or hybrid systems blending organoids with silicon for fault-tolerant AI that learns like a child, fostering innovations from self-driving cars to neural prosthetics. As of October 4th, 2025, the latest developments have mini-brain computing surging ahead, with FinalSpark's announcement on September 30th of their neural platform going public for remote access, allowing global researchers to train organoid networks for tasks like speech recognition, with early tests showing 10 times the efficiency of GPU-based models. Just yesterday, October 3rd, a Johns Hopkins team published results on a vascularized organoid that sustains activity for 120 days, a leap from the previous 60-day limit, enabling longer training sessions for complex computations like protein folding. Ethical debates heated up on October 2nd, with a Nature editorial calling for international standards on organoid sentience, amid Swiss regulators approving FinalSpark's 16-organoid biocomputer for commercial trials. No major breakthroughs in scaling to full computers yet, but momentum's building, with U.S. DARPA funding similar projects for energy-efficient AI in defense applications. Mini-human brains powering computers are a biotech fusion that's efficient, adaptive, and ethically charged, promising a greener path to smarter machines. What are your thoughts on organoid computing? Are you excited about the energy savings, concerned about the ethics, or curious about its medical potential? Share in the comments below, like if this sparked your curiosity, share with fellow tech fans, and subscribe for more deep dives into science's bold frontiers. Thanks for watching. See you next time.